We have a very comprehensive program today, and we're going to start out the program with our distinguished neurologists. The first one being Dr. Larry Golbe, who serves as CurePSP's Director of Research and Clinical Affairs. Larry's been with CurePSP since the very beginning, since 1990. He's chair of our Scientific Advisory Board and also a member of our Board of Directors. Larry is a professor of neurology at the Robert Wood Johnson Medical School in New Brunswick, New Jersey, and has specialties in Parkinson's disease and movement disorders. His research is in the genetics and epidemiology of Parkinson's disease and related disorders, and he led the clinical portion of a project that identified the first known gene for Parkinson's disease. His work in PSP has centered on the genetics and epidemiology, and he devised the PSP rating scale, which has become the most widely used clinical measure for PSP worldwide. And we're very grateful for Larry's service to the Foundation for these many years. Larry?
Thank you, Richard, and thank you all for coming out here today. As I learned when a couple of my colleagues and I took a drive from this spot to Fort Worth to be interviewed on the local NBC station the other day about PSP, I learned that there really are wide open spaces here in Texas. So I appreciate, and coming from New Jersey, it's really quite a shock. So I know that many of you probably had to travel some distance. And the traffic is just as bad as in New Jersey. So I hope you didn't come to this lecture thinking that I was going to tell you the causes of PSP, because we don't know yet what the causes are. But the point to be taken home from this slide is that it's not one cause. It's going to be multiple causes. PSP, like most of the neurodegenerative diseases, is probably going to turn out to be more than one disease that just kind of looks superficially similar. Of course, it depends on what you define as a disease. Now, it's considered customary in my business to reveal the outside organizations that provide money for my research and stuff, and this is the list. And I'm grateful to all of these organizations. That's so that in case I might have a conflict of interest. Now, what do we mean by a cause of a disease? Well, there's two parts to a cause. There's this stuff acting on your body from outside, that's called the etiology, and then there's what happens inside your body as a reaction to it, the series of events that actually cause the symptoms and disability. So etiologies of diseases can be things like toxins from environment, food, medication, or radiation, like Pat mentioned. They could be genes, which come from your parents, that's outside your body. Trauma, infectious agents, or psychological stressors are also from outside of you. Then the pathogenesis is processes, for example, inflammation, buildup of atherosclerotic plaque in your arteries, insufficient level of some hormone, or loss of specific cells and specific organs, and that's the kind that we're talking about here today. But I could have made these lists a lot longer. I just wanted to give you an idea of what we're talking about. Now, Dudley Moore asked not to be the poster boy for PSP, but that's what he is anyway. Or at least during his life he wasn't, but after death. He's somebody who is a well-known person he's not as famous as Michael J. Fox, but still, it's amazing how a celebrity who has the disease really, it helps people who have the disease kind of feel like they're not alone. And it's also good for increasing awareness in the general population. These are some less famous people who had PSP, some of my patients. Notice it's called the over-innervated face. The facial muscles are contracted more than normal. I kind of think of it as somebody who's just smelled something bad. Not everybody has this kind of expression on their face, of course. The main features of PSP, I'll just start this kind of clinical description very briefly. I think this is my only slide on that. Usually starts with balance problems. And within the next few years, it affects these things, not in this order necessarily. Speech, swallowing, slowness of movement and thought. This is an insufficient movement. These two things are called Parkinsonism, and that's why PSP is one of the Parkinsonian disorders. And it's called one of the atypical Parkinsonian disorders because it has more balance problems than Parkinson's disease, and it has more eye movement problems, and it has more of this stuff, frontal behavior and other personality change from the frontal lobe of the brain, reduced bladder control more so than in Parkinson's, and more sleep difficulty than Parkinson's. Now, PSP pathology, well, here what we're talking about is the typical appearance of the brain under the microscope. It's not a one-to-one correspondence of a classical outward picture of PSP with the microscopic appearance through the microscope. People with classic PSP pathology can have a number of different variations. The classic one is called Richardson syndrome, because Dr. Richardson was one of the 3 doctors who originally described the disease in 1963. But there are these other variants. One looks a little more like Parkinson's disease. That's a third of people. And about 12% have some combination of the top 2. And then there are small numbers that have these other things as the predominant feature. Pure akinesia with gait freezing. That just means a very great difficulty moving, great difficulty initiating the first step. Corticobasal syndrome is very asymmetric. It's much more on one side of the body than the other. About half the people who have corticobasal syndrome end up having corticobasal degeneration. And the next slide will talk about some of these complications. Apraxia of speech is a particular kind of speech difficulty that occurs with other types of pathologies as well. This shows these overlapping rectangles give you an idea. I won't try to explain all this in detail here, especially because it's hard to see. This gives you an idea of how PSP is not just one disease. It's a spectrum in several dimensions. It overlaps with other disease. The overlap occurs not only at the clinical level, in other words, the outward signs and symptoms, but also at the pathological level, the appearance through the microscope. So in other words, it's a very blurry kind of picture. And in that picture, the pink ones are the clinical pictures, and the green, blue, and purple are the pathology through the microscope. So you see PSP pathology is this rectangle here, and it overlaps with a lot of different clinical pictures. Corticobasal degeneration pathology can have a number of different clinical pictures, and it overlaps with other pathologies. PID, this means Pick's disease. That's a type of frontotemporal dementia. And post-encephalitic Parkinsonism is a type of Parkinsonism that doesn't occur anymore, but it was extremely common back in the early 20th century as a result of an epidemic of a viral infection of the brain. After people recovered from the encephalitis, their coma, they would wake up, and within days to years after that, they would come down with a Parkinsonian disorder that did not look outwardly like PSP, but through the microscope, it looks exactly like PSP. So that gives you a good idea of how there's so much lack of sharp focus of these diseases. Now, let's get down to the nitty-gritty of the cause of the disease. This is in the pathogenesis. Here are these neurofibrillary tangles in the brain. Each of these black blobs is a neurofibrillary tangle. These are made of a protein called tau, T-A-U. Here's a higher power view of one that's in an astrocyte, which is these are cells in the brain that are not electrically active. These are the cells that supply the nutrients and other things for the cells that are electrically active. And in fact, PSP may start in those non-electrically active cells and then spread to the electrically active ones, which are called neurons. And here's a kind of a cartoon of what's going on. Here's a healthy brain cell. It's got its axon coming out. That's the long wire, the cable that comes out of it to connect to other brain cells. And it's like a coaxial cable. It has these microtubules inside the cable. They're kind of like, think of them as kind of like the bones, the two bones in your forearm. The microtubules act as an internal support for the axon, for the long cable. And each of these microtubules, if you magnify it, is a spiral-shaped stack of two other proteins called tubulin and actin, not important right now. But that stack would break down if not for these little scaffolding things that are arranged on the outside of it. And those scaffolding things are protein molecules of tau. That's the normal function of the tau protein in our bodies. We need it. Here's a brain cell that's breaking down, and here we don't know why it's breaking down, but it's breaking down. The microtubules are coming apart, the tau is falling off, and the tau is forming these clumps. Now, we don't know which is the chicken and which is the egg. Is it that the microtubule breaks down first, and that frees up the tau, and the tau is just floating around with nothing to do, so it starts congregating on street corners like any other unemployed, teenagers, and getting into trouble, causing trouble? Maybe that's it. Or maybe there's something wrong with the tau, and that makes them not work well. And then, since they don't work well, that allows the microtubules to fall apart. We don't know which is the chicken and which is the egg yet. Here is a somewhat more sophisticated version with some other details. The point of this slide is, see, here's the tau attached to the microtubule. The point of this slide is that the tau has you can't see it, these P things. P. That's phosphate. Phosphate groups, it's a little chemical, gets attached to the tau in abnormal places, abnormal amounts, and that affects the behavior of tau. It's a normal thing for tau and many other proteins to get phosphate attached. It's part of our body's way of regulating the behavior of proteins. It's a normal thing. But like any other normal thing, it can be overdone. It could be done in the wrong place. And that's what's happening in PSP. There's over-phosphorylation of tau. Again, we don't know if it's a chicken or an egg. We don't know if it gets over-phosphorylated because the tau's misbehaving and the excess phosphorylation is the body's way of starting to break down the tau. Or maybe the problem is the over-phosphorylation to begin with, which impairs the functioning of the tau. And then once the tau is over-phosphorylated, it tends to aggregate together, and it should be broken down, but it isn't broken down enough, and that can cause problems. And here's how it causes problems. This is another step in the pathogenesis. Still with me, everybody? Yeah. All right. Here is a healthy brain cell with its axon dendrites. Those are the connections to the other brain cells. And here is the brain cell with these aggregates of tau that are starting. And eventually the aggregates of tau, I won't say they kill the cell, but they are liberated into the fluid when the cell dies, and then they can be taken up again by other healthy neurons in this vicious cycle. And this slide shows potential areas of research that may try to either detect, diagnose, or interrupt this vicious cycle. So biomarkers, that means a diagnostic tool to help us tell that this process is going on early in the game, while we still might be able to do something about it. Biomarkers are what we're looking for in PSP. And this says small molecule inhibitors, in other words, drugs that you can take by mouth and will get into the brain when taken by mouth that hopefully can prevent various stages of this process. And this says immunotherapeutic inhibition, in other words, a vaccine harnessing the body's immune system to maybe attack the abnormal tau aggregates and prevent them from killing brain cells and getting into other brain cells. All right, so that's what those are among the things that we're trying to do, and you'll hear more about treatment of PSP later. Now, I want to just, before we proceed any further, I just want to list all the things, or most of the things, that contribute to the cause of PSP. I've got 4 slides on this, so I'll go through this pretty quickly. Number 1, causes of tau misbehavior that are not directly genetic. Misfolding of the tau molecule, possibly related to abnormal attachment of those phosphate groups that I showed you. Aggregation of tau into groups of 20 or 30 tau molecules at a time. Those are called oligomers. Oligo is Greek for few. Tau oligomer toxicity. It's thought that the toxic form of tau is the oligomers and not the larger aggregates, which you can see through the microscope, called neurofibrillary tangles. Extracellular transmission of misfolded oligomeric tau. That's what I showed you in the previous slide, down here, from cell to cell. Excess 4-repeat tau, I'll explain what that means in a minute. And abnormal modification of the tau protein by other things that are not genetically dictated, like the actual structure of the tau protein is genetically programmed. This stuff is not. Phosphorylation, attachment of phosphate, attachment of a sugar, attachment of something called ubiquitin, which is important for the breakdown of abnormal proteins, attachment of a sulfate group, and oxidation of proteins. These are all things that happen normally every day to our proteins, but they happen in an abnormal way to the tau protein in PSP. Contributors to the cause of PSP continuing. Genetic defects in tau. Mendelian versus non-Mendelian. What do we mean by that? Well, you remember Gregor Mendel and his green pea plants, where he found that tallness or shortness is inherited in a very crisp, easily measured way. Well, that's called Mendelian genetics, and that's what happens when you have just one gene controlling the trait. It's easy to trace the effect of that gene down through the generations. So something like Huntington's disease is a Mendelian disease because it's one gene, and if you have the gene, you have the disease, and if you don't have the gene, you don't have the disease. That's Mendelian. That's the simpler kind of genetics. There are a few rare families who have PSP that behave like that, but most of the genetic influence on PSP is non-Mendelian. In other words, it's going to be multiple genes, each of which has a small contribution, and you would need more than one of these genes in the same person to cause the disease. Well, we're gradually learning about these genes that each give a small contribution. 2 of them are in tau. 2 of them that we know of are in the tau gene. One is called the H1 haplotype, and the other is just a single substitution of one letter in the genetic code. The H1 haplotype is not a single substitution of one letter. The H1 haplotype is where a whole section of the tau gene, a whole section of chromosome 17 that includes something like 6 or 7 genes, including the tau gene, where a whole section is just reversed in order in its positioning on the chromosome. So when you're reading the code on chromosome 17, suddenly you reach a section that's reversed. That's the tau H1 haplotype. Now, the whole genome analysis that CurePSP has recently sponsored And thank you to all the donors who gave money for that project. It was about $1 million project, and it was just published last year. There were 3 genes that emerged from that. These are not Mendelian. These are acting in the non-Mendelian manner, each of them contributing just a small amount. I'll tell you, I'll describe what these are doing on subsequent slides. And a handful of genes related to the inflammatory response, in other words, the immune system. And that seems to be contributing to PSP as well. Now, other contributors to the cause of PSP, problems with the mitochondria. I'll talk about the mitochondria in a minute. I'm just cataloging all these things for you first before we plunge in more detail. The mitochondria are the little factories in our cells that make energy using oxygen and our food, sugar. They're going wrong in PSP. And toxins. We don't know what the toxins are, as Pat said, but we have a couple of little clues, and I'll get to those later. Now, something else going wrong in PSP has to do with the tau gene, and it's probably not a problem of genetic origin, but we don't really know yet. It has to do, this is getting a little technical now. This is as technical as I'm going to get. So if you get through this slide and maybe the next one, it's downhill from there. Here is a schematic view of the tau gene. Now, every gene can be broken up into its exons. The exons are the parts of the gene that actually get translated into protein. The places between the exons, which are called the introns, we don't know what that's doing. It may just be junk DNA left over from millions of years of evolution. It's been turned off and it doesn't do much anymore. Or it may have certain functions. Some functions of the introns are being discovered more and more. Anyway, you may have read recently that the tomato plant has 32,000 genes. This was just discovered. While humans, we only have 25,000. Does that mean that we're simpler creatures than a tomato plant? Well, the answer is no, but we do things differently than the tomato plant. In order to accomplish all the tasks we need to accomplish, we did not just make new genes through millions of years of evolution when we needed something new to happen. What we did, in many cases, is take an existing gene and just pick and choose from the different exons to make different versions of the protein. So in the tau gene, this is called alternative splicing. Some of the exons are optional. In the case of tau, exons 2, 3, and 10 are optional. And this allows us to make 6 different forms of the tau protein, normally in our brains, or in our bodies. There are 6 different forms. 3 of these 6 forms, now notice you can have exon 2 without exon 3, but you can't have exon 3 without exon 2. And you can have 10 either in or out. Now, exon 10, this one is special because part of the protein that comes from exons 9, 10, 11, and 12 has what's called a microtubule binding domain. Remember in one of the first slides, it showed the microtubule, that cylindrical tube that had the tau molecules hanging on the outside. Well, the tau has special places in its structure where it sticks to the microtubules. It has either 3 or 4 such places where it can stick to the microtubules. And in normal human brain, about half the tau molecules have 3 microtubule binding domains because they do not have exon 10. And the other half have 4 because they do have exon 10. It's about 50-50 in the normal human brain. But in PSP, most of the tau molecules in the cells that are getting sick have 4 repeats, 4 microtubule binding domains. In other words, they have exon 10 that fails to get spliced out. So a failure of splicing out of exon 10 seems to be part of the pathogenesis of PSP. Now, there are other diseases. This is a list of most of the tau disorders. Corticobasal degeneration also is mostly 4 repeat tau. Frontotemporal dementia, some forms of it. Guadalupean Parkinsonism, which I'll talk about later. And this rare disease called argyrophilic grain disease also have 4. There are a couple that have mostly 3, and there are some that, like the normal brain, have equal amounts of 3 and 4, and one of them is Alzheimer's disease. Another one is post-encephalitic Parkinsonism, and another is some forms of frontotemporal dementia, and another one is this tauopathy that occurs only on the island of Guam and some neighboring places in the Western Pacific. Now, the thing is, this is still a chicken and egg problem. We don't know what is causing this change in the ratio of 4 repeat to 3 repeat tau. And we don't know what the mischief is that the excess of 4 repeat tau is doing. So the neurofibrillary tangles in PSP have an excess of 4 repeat tau, but the significance of that remains unclear. All right, so let's talk more about genetics. Now, when my patients ask me, is my PSP, am I going to pass this down to my children? The answer is no. It is not a familial disease. It does not occur measurably more often in families and in a patient's family than it does in anybody else. Maybe if you took 10,000 people, then maybe you'd see a slight difference. But it's less than 1% of people with PSP has somebody else in the family with PSP. However, there's always a however. If you look very carefully at the relatives of people with PSP and you give them a test of their reaction time, you find that the relatives of people with PSP tend to have slower reaction time than the relatives of people without PSP. And if you give a battery of tests that test other things like sense of smell, emotional reactions, other movement things, again, there's a difference between the relatives of people with PSP and without. They didn't have PSP. They were probably never going to develop PSP in their lifetime. Maybe if they lived to the age of 150, maybe then they would develop PSP. But for practical point of view, it's not a familial disease. And there are 2 other studies that show a failure to demonstrate a familial clustering. This looks at a family history of either dementia or motor Parkinsonism. In other words, these researchers said, well, okay, if PSP itself isn't happening in the relatives of people with PSP, but let's look at things, some other Parkinsonian features, things that are a little too subtle at this point to be diagnosed as PSP. They found that in either PSP or corticobasal syndrome, there was a family history of such things in 32%. In the controls, there was a family history in only 22%. But it did not reach statistical significance. In other words, it could have been a fluke. In order for it to be considered not a fluke in medical research, this value, this p-value, has to be less than 0.05. In other words, less than a 1 chance out of 20 that it's a fluke in order for us to take it seriously. And this did not meet that standard. And a similar but more sophisticated study was done in Europe where they looked at incidence of Parkinsonism or dementia, similar to the previous one, in relatives. And here they plotted it along the course of their life. This on the left is MSA, so don't pay attention to that. On the right is PSP. The top right is incidence of Parkinsonism among the relatives, and the bottom right is the incidence of dementia among the relatives of people with PSP which is the solid line, and among the relatives of controls, which is the dotted line. And you see the solid line and the dotted line are more or less following the same path. If anything, the controls are a tiny bit worse, but that certainly did not reach statistical significance. So in other words, this is another negative study showing no signs of familial clustering of PSP. Nevertheless, there can be a genetic contribution to the cause of PSP. How can that be? Well, because maybe multiple genes are necessary in the same person. Maybe you have to inherit 3 abnormal versions of a gene from one parent and 2 from the other parent. I mean, maybe you need 5 altogether, and you can get 3 from one parent and 2 from another parent. Neither parent has it. Probably none of your siblings inherited the same combination, unless you have, 100 siblings. So that could obscure a genetic component. Recessive inheritance, that's where you have to get the same abnormal gene from both parents. You may have to have not only a genetic etiology, but also an environmental etiology in the same person. Older age onset. We say that the average age of onset of PSP is 63, but maybe somebody in a family would be 83 when they developed it, except they died of a heart attack at 75. Well, then you'll say, well, that person didn't have PSP. Non-paternity, 1% of the population. Mama was fooling around. And stochastic factors. What is a stochastic factor? This is kind of a difficult thing for people to get their heads around, but I know you're willing to give it a shot. A stochastic process. This is where there is some randomness going on. And in order for the event of interest to take place, the random events have to pass some threshold, just by chance. They pass some threshold, and then a whole other series of events is set in motion. It's like triggering. It's kind of like if you took a gun. I just thought of this analogy. If you took a gun and you're kind of fooling around randomly with the trigger, and sooner or later you pull the trigger enough that the gun fires. That's a stochastic process. Or if you're heating up a saucepan of marinara sauce, and it's bubbling away, and the droplets are mostly falling back into the pan, but once in a while you turn up the heat enough, and now the droplets, randomly one will go on your shirt. And if you turn up the heat further, more will go on your shirt because you've increased a certain initial condition. That's a stochastic process. And that may be happening in PSP and all of the other neurodegenerative diseases, but instead of pressure on a trigger or bubbling of marinara sauce, instead it's folding of the tau molecule. And maybe the abnormal folding of the tau molecule is made more likely, maybe by abnormal phosphorylation, maybe by exposure to some toxin, maybe by something else going on in the cell. It's just happening randomly, but maybe it's happening more often as a result of whatever is going to turn out to be the cause of PSP. And then it produces a self-sustaining chain reaction of protein misfolding and cell damage. You remember that triangular diagram I showed you before, with release of the growing number of misfolded tau molecules into the spaces between the brain cells and subsequent uptake by normal brain cells. Chain reaction spreads along pathways in the brain. All right, so one of these genetic influences, the H1 haplotype, I mentioned that one. That's where the section of the chromosome 17, including the tau gene, is reversed. How frequent is that? Well, if you look at all chromosomes in the population, each person has 2 chromosomes. About 94% of 94% of the chromosomes in people with PSP are of the H1 flavor, and similar with CBD, but only 77% of controls. Well, now you think, well, that's not that impressive a difference, 94% versus 77%, but that is a very big difference when you're talking about non-Mendelian genetics. And if you look at individual people, where you tally up how many people with PSP have both of their copies of chromosome 17, both the maternal and the paternal copy, both of them are of the H1 flavor, 88% in PSP, 60% in controls, starting to look a little more impressive. So the H1H1 genotype, the genotype means the combination of the genes from both parents. The H1H1 genotype is nearly necessary but far from sufficient for PSP to develop. And if you have one chromosome 17 with the opposite flavor, the H2 haplotype, it reduces your PSP risk. If you have both chromosome 17s with H2, it reduces your risk even more. But what conditions the genetic effect? We don't know. And this was the purpose of this whole genome association project that just got finished last year and published, which was a huge deal for our organization. We had a researcher at the University of Pennsylvania, Jerry Schellenberg, organized this consortium of geneticists. And he was assisted in this by Gunther Herglinger, who's in Germany. Gunther convened the guys in Europe, and Jerry did so in North America. And it was published in Nature Genetics, which is the most prestigious genetics journal. So here is the actual published summary abstract that appeared in the journal. And I'll just explain this so you understand it. PSP is a movement disorder with prominent tau pathology. Brain diseases with abnormal tau deposits are called tauopathies. You may think this is kind of simplistically explained, but keep in mind that the readers of Nature Genetics don't necessarily know much about PSP. They may be specialists in tomato genetics or something. The most common tauopathy is Alzheimer's. Environmental causes of tauopathies, some tauopathies include repetitive head trauma associated with some sports. The authors threw that little statement in there in order to make this look more relevant to the world in general and increase the chances of acceptance by this prestigious journal. But it's still, that's a very important point. So to identify common genetic variation contributing to the risk for tauopathies, we used about 1,100 PSP cases. These are samples of DNA taken from brains in our PSP brain bank at the Mayo Clinic in Jacksonville. And 3,000 controls. These were DNA samples taken from healthy children, not healthy children, but children going to the Children's Hospital of Pennsylvania for various problems, any kind of problems, obviously not PSP. And yeah, maybe a tiny percentage of them would eventually develop PSP when they were adults, but few enough that we could count them as controls. Followed by a second stage where there were 1,000 cases, many of which were subsequent autopsy cases that we had accrued since the first stage, and another set of controls. They were genotyped, in other words, their genes were analyzed. For single nucleotide polymorphism, that means a single letter in the genetic code is substituted. That yielded a p-value of less than 10 to the minus 3. In other words, that's 1 out of 1,000. Remember I said that it has to be less than 1 out of 20 to be considered a finding. We insisted on less than 1 in 1,000. And as you'll see, the most impressive results were way less than that. We found these three associated with less than 5 times 10, less than 1 in, I don't know, what is that, 100 million. For these 3 genes, STX6, EIF2AK3, and MOBP. I'll explain what these are in a second. And we confirmed 2 independent variants in tau gene, one of which affects tau brain expression. MAPT means tau. Microtubule associated protein tau. So the conclusions, the 3 newly implicated genes encode proteins for, this is what these 3 cryptic genes mean, a vesicle membrane fusion at the Golgi endosomal interface. Okay, everybody cool with that? I'll explain what that means in a second. The endoplasmic reticulum unfolded protein response, and a myelin structural component, plus 2 defects in tau, one of which is the H1 haplotype was already known. It's been known since about 1998. This is the data. Don't worry about these numbers. The point of this slide is to show you the p-values here. The one for the H1 haplotype, 10 to the minus 116th. There isn't a name for that number, but that's the chance that this was just a fluke. 1 in 10 to the 116th. Even the weakest of these was 10 to the minus 10th, which is 10 billion. 1 in 10 billion chance it was a fluke. Okay, so here's what all that gobbledygook means. There are various environmental stresses like heat and ultraviolet radiation that are constantly trying to damage our cells. And one of the ways that cells deal with that is this very complicated mechanism. And I certainly won't get into the details, but part of that mechanism is this protein called the eukaryotic translation initiation factor 2. This is the nucleus of a cell under the electron microscope, and these kind of tubes around here are called the endoplasmic reticulum, the ER, and here's a artist's cutaway view of it, and this is the reactions that are happening in that area. So this means that one of these genes that are acting in a subtle way in PSP has to do with the ability of the cells to deal with insults, with physical type of insults to the cell. Another one is this STX6, has to do with the organization of these vesicles. Vesicles are these little bubbles with chemical in them that eventually will fuse with the outer membrane of the cell and release their contents into the space here. And the cells do this for various reasons. One is to get rid of garbage, and one is for neurotransmission, so that our brain cells can talk to each other using these little pulses of chemicals. And similarly, brain cells can take in little blebs of the external membrane, pinch them off, and make new vesicles. And this is a critical process for cells, and STS6 is one of the proteins involved in doing that. The third one, MOBP, is one component of myelin. Now, what is myelin? Myelin is what makes our white matter white. It serves as the insulation around the axons. Remember, the axons are these long cables coming out of our brain cells. A lot of them have insulation so that they conduct better and faster. And the insulation is made mostly of myelin, and MOBP is one of the genes that codes for a relatively minor component of the myelin. When your mitochondria start to get damaged, they leak their contents into the rest of the cell. And one of the things they've got in there is apoptotic factors. Apoptotic factors are chemicals that set in motion the cell's suicide machine. In our cells, they have a program that is waiting, that is not in action, but is potentially in action, that kills the cell, that sets in motion stuff that just results in the cell's death. And this is useful if you're, say, a developing fetus, and you grow certain tissues, and you have to reabsorb those tissues in order to allow the permanent version to grow. And there are things the adult uses it for as well, and it can be set in motion abnormally if your mitochondria are leaky, and that is probably part of the pathogenesis of PSP. So what's going wrong with the mitochondria in PSP? There may be a poorly characterized genetic defect, and we have evidence for a genetic defect in mitochondria in PSP. Impairment of complex 1, which is part of that's one of the proteins involved in extracting energy from oxygen and sugar, maybe on a genetic basis. And the oligomers of tau, those small aggregates of tau, may damage the mitochondrial membrane. Now, when these proteins like tau aggregate, the cell tries to get rid of them. Even when they misfold, the cell tries to get rid of them. There's a normal mechanism in the cells for getting rid of misfolded proteins or worn out proteins or just overproduced proteins. That's called the proteasome. It's like a little garbage disposal inside the cell. And just like a garbage disposal, the garbage goes in the top and it gets chewed up in the middle and it comes out the bottom. And this is what it would look like if you were at this point looking down into it. This is the hole through the middle. And the problem in one problem in neurodegenerative diseases seems to be that these garbage disposals get clogged. They just get overwhelmed. So here's misfolding of a protein that overwhelms its garbage disposal mechanism. This is a very particular kind of protein that is not a problem in PSP, but it's a problem in a bunch of other diseases that are very severe diseases of the brain called the prion diseases. And this is a prion protein. We don't know what the normal prion protein's normal job is, but we do know that it misfolds sometimes, and when it misfolds, it gets other normal prion proteins to misfold, and then they aggregate and they turn into clumps, and they get transmitted from cell to cell in this way. The prion diseases are things like mad cow disease, Creutzfeldt-Jakob disease, bad diseases. And it may be that PSP and Alzheimer's and Parkinson's operate by a similar kind of mechanism. Here is a plaque of prion protein in the brain. So let's move on to something a little easier to understand, the environment. We don't know much about the environmental insults. Pat listed a few candidates, but we really don't know. Lesser educational attainment was found in 2 studies, one of which I did. Slightly less education in people with PSP. How might this cause PSP? Well, maybe people with less education are more likely to be exposed to, factory-related toxins, farming-related toxins, as opposed to people who have office jobs. Maybe people with lesser education are more likely to live in the part of town where there's more toxins. Maybe there's a difference in the diet between people who earn more money and earn less money. Maybe there's less synaptic reserve, where education allows you to build more connections between your brain cells so that then later in life, when you come down with a neurodegenerative disease, it doesn't show symptoms so early. What is the evidence that there's a dietary cause of PSP? Well, on the island of Guadeloupe in the Caribbean, there is a increased, there is a dietary use of these fruits, and the people on that island who use the fruits are more likely to come down with a PSP-like illness. There's another disease similar on the island of Guam, a PSP-like illness. We really have no clue what the cause of that is. It's been studied now for 50 years, and we're still in the dark. All sorts of theories have been proposed and discounted. Here's Guadeloupe, beautiful place. Here's soursop and sweet sop. It's available commercially, including in the Caribbean restaurant around the corner from my office. And this shows the statistical association of PSP relative to people on Guadeloupe with Parkinson's disease. The PSPs were more likely to have these fruits in their diet than either people with Parkinson's or controls on Guadeloupe. My conclusions. PSP does not clearly cluster in families, but perhaps early, mild, inapparent symptoms are more common among the relatives of those with PSP, perhaps. Mutations acting in Mendelian fashion are rare in PSP, but mutations acting in non-Mendelian fashion as genetic risk factors account for a minority of the overall cause and are easily identified. There are many possible defects in tau, including attachment of smaller chemicals like phosphate, misfolding, aggregation, insufficient disposal by the proteasome, and the abnormal ratio of 4 repeat to 3 repeat forms. Is all this flashing past your eyes again? Other likely contributors, mitochondrial problems, toxins, prion-like misfolding with aggregation, cell death, uptake in this vicious cycle, And the remaining big questions, which are the chickens and which are the eggs, which are most susceptible to a treatment, and will the answers for PSP be the same as for the other neurodegenerative diseases and vice versa? There's obviously a lot more work going on in these things than PSP, and we hope that whatever is found for these will help for PSP, and we certainly hope the opposite as well, and time will tell. Thank you for your attention.